Now we're going to start discussing DNA replication. So here we're starting our walk through the central dogma of molecular biology with the top round arrow there for DNA replication. This is chapter 12 in your textbook. First, we'll discuss a couple of experiments that were important in determining how replication works, and then we'll talk about replication in prokaryotes, then different structures that the DNA molecules undergo as they replicate, and then lastly we'll finish with eukaryotic DNA replication. So this is an install. In 1958, grew E. coli in a heavy isotope of nitrogen and 15 for several generations. Then I transferred it to the more common N14 and sampled the DNA at various times using density gradient centrifugation, and that made them be able to distinguish between the different DNAs made with the different nitrogen isotopes. And after a single generation, they found an intermediate band that contained one strand of heavy nitrogen, one strand of regular N14. And this led them to conclude or discover that DNA replication is semi-conservative. As we can see there, one strand is conserved and one new strand is synthesized each time the molecule is replicated. So this shows that when you spin it down, you've got N14 DNA and N15 DNA. You can distinguish them in the test tube. And after a single generation, they have, as you can see in the second from the left there, one band that's intermediate between the light and the heavy nitrogen. And so this shows you the semi-conservative nature of DNA replication with the molecule with the yellow sugar phosphate backbone, the new ones, and the old ones being the templates, those with the bluish sugar phosphate backbone. The next second experiment, a guy named Cairns in 1963 used autoradiography, and that's defined as using radioactivity to expose X-ray film. So he grew E. coli in media with radioactive thymine, so every time there was a T incorporated into the new DNA strand, it was radioactive. And from that, he made three important discoveries. One is that the DNA molecule in prokaryotes is circular. Secondly, the molecule retains its integrity. During replication, it doesn't break apart and reform, and it forms this theta structure, like the Greek letter theta. The replication occurs simultaneously in opposite directions at two wide junctions. That was his third discovery. So DNA and RNA are both polymers, long strings of smaller molecular entities called monomers. And so we have to start the process with an initiation phase. And the elongation phase is merely adding more monomers. And then somehow we have to stop the process with a phase of termination. So the origin, and this is mostly for E. coli, the ori is a 245 base pair region where replication begins. Replicon is defined as a length of DNA replicated after a single origin. Viruses and prokaryotes only have a single origin. Yeast, which is a unicellular eukaryote, has about 500 origins. Mammals can have up to 60,000 origins, because we have pretty large genomes. And with different linear chromosomes, there would have to be at least one origin per chromosome, but there really is many. This shows right from your textbook the number of replicons, as well as how many bases are replicated in between those. So about 40,000 for yeast and fruit flies. You know, fruit flies have way more origins. 200,000 for the xenopus, the African clawed frog, and about 100,000, 150,000 in the mouse. In between their number of origins of replication. This shows what it looks like in the E. coli chromosome. You can see there that they tend to be AT-rich regions. Remember, AT base pairs have only two hydrogen bonds holding them, so in areas where the DNA has to unravel a lot, you see those tend to be AT-rich regions. Now the ends mean that can be any of the four bases there, but T is for thymine, of course, C for cytosine, A for adenine, etc. So what happens is initiator proteins bind to the ORI. As we can see here, it's lighter than the DNA on either side of it, and wrap around it. And then right next to that, the DNA begins to unwind. And these single-stranded binding proteins are go in there and hold it open. Think of them as like little clamps, because of course naturally the DNA wants to twist around. It doesn't want to be straight. So think about it kind of like a metal spring or something. So here we can see molecules called DNA helicases and DNA gyrases are important in unwinding the DNA. And again, those single-stranded binding proteins hold the DNA straight for long enough where it can be copied. And of course, this process is controlled by enzymes. DNA polymerases are enzymes that make DNA. DNA polymerase 1 fills the gaps in prokaryotics, DNA replication. DNA polymerase 2, responsible for repairing damaged DNA from external sources. DNA polymerase 3 is our main enzyme for making the new strand, so that's going to be the one we talk about the most here. Primase makes primers, which are RNA nucleotides. The helicase is involved in unwinding the DNA. We really won't talk much about RNA polymerase 2 here. And this just lists the prokaryotic DNA polymerases and what they do. So what happens at the origin? Well, the initiator proteins bind there. However, if the cell does not need to copy its DNA, then there's an off switch, which are proteins that bind to the ORI, and they block out the initiator proteins, and that prevents DNA replication from occurring. So that's how cells turn off replication when they don't need it. So anyway, helicases and gyrases come in and start unwinding the DNA after the initiator proteins attach. Primase lays down these RNA primers, and then DNA replication occurs simultaneously in both directions away from the origin. So here we see the origin there in yellow. And then if we zoom in on that, we can see the beginning of a small replication bubble, and those small arrows show the directions of replication with the new DNA in red there. And then later on in the process, we're about halfway through the circle on the right there. This shows you what Cannon saw in his autoradiographs. So here you can see a small replication bubble on the left, the theta structure in the other three, and on the right there, replication is almost complete. And this shows you the last couple slides together as one slide. And the DNA replication stops when the two Y junctions meet at the bottom of the molecule or the opposite side of the origin. So early on, we've got molecular complexes with helicases, primases, and gyrases all working to unwind the DNA and lay down the primers. So those green things are RNA primers that the DNA polymerase will come and add more nucleotides to. And this shows that the DNA then has to be unwound with the new strands being made in yellow here on the inside. Also, it shows that DNA polymerases can only add new nucleotides at the 3' end of a growing chain there. Also that the nucleotides exist as triphosphates in the cell when they're not incorporated into the DNA or RNA. So, the polymerases need that 3' OH to attach a new nucleotide to. And so new strand synthesis can occur in the 5' to 3' end direction, only. And so what's called the leading strand is the new strand where the 3' OH is always exposed, and DNA polymerases can just continue adding nucleotides to that. But the lagging strand is synthesized discontinuously, so it's made in such a way that the 5' end is exposed, and therefore the new strand has to be synthesized backwards relative to the way the molecule is unwinding. And we'll talk about that shortly here. So this lagging strand is made in these small fragments called Okazaki fragments. And the steps used in making those First is primer synthesis, so that's anywhere from 2 to 60 base pairs of RNA laid down by the enzyme primase. Then DNA polymerase comes in, adds new nucleotides to the 3' end until it runs out of room, reaching the previous Okazaki fragment. Then DNA polymerase 1 comes in, removes the RNA primer, and fills the gap with DNA nucleotides. And then lastly, there will be a gap left, a nick between 2 adjacent nucleotides in the sugar phosphate backbone, and DNA ligase will come in and close that by a process called ligation. So here we see down towards the bottom of the slide, the leading strand being synthesized continuously, and then the smaller Okazaki fragments of the lagging strand. This is primase coming in and laying down RNA primer so that there'll be a 3' OH available for DNA polymerase to work from. To show some of the steps in lagging strand synthesis. So the arrows point out initiation sites. The purple nucleotides there are the primers. And then in step 3 there along comes DNA polymerase 3, adding DNA nucleotides in green there until it reaches the previous Okazaki fragment. And then at 4B, DNA polymerase 1 has come in, plucked out the primers and replaced them with DNA nucleotides. And then in 5, we have DNA ligase closing up the nicks, the little gaps in the sugar phosphate backbone. And that shows you a nick there and DNA ligase closing that up. And so this shows, again, primer removal and gap filling by DNA polymerase 1. So we have to have that 3' OH for DNA polymerases to add new nucleotides to. And this shows primer removal and gap filling at the top, and then ligation at the bottom. So here's what a nick looks like up close. It's just a gap in the sugar phosphate backbone between 2 adjacent nucleotides. The P's there are the phosphates. The things that look like rungs of the ladder are the base pairs, of course. And then DNA ligase comes in and just plucks out the phosphate and creates a new bond that way. Now the Y junctions are also called replication forks. The molecule has a theta structure to it. At each Y junction, we have 2 DNA polymerase molecules, DNA polymerase 3 molecules. The leading strand is fed directly into the DNA polymerase 3. The lagging strand is unwound for about 1,500 base pairs and held straight by the single-stranded binding proteins. And then, of course, DNA polymerase 1 and DNA ligase do what we just talked about. So this shows you with leading strand on the left, lagging strand on the right. Primers in red, single-stranded binding proteins as those things that look like little green seeds or something. And here's a close-up of the replication fork with the DNA polymerase 3 molecules and the DNA being fed through them. Now, termination. When the Y junctions meet, an enzyme called topoisomerase releases both circles. DNA polymerase 1 and DNA ligase close the circles up, and now we've got 2 new DNA molecules. So here the new strand is in red and the old one is in black. So on the left there, it's just about to be terminated, and at the right, it has become terminated. DNA replication is over. Now, DNA polymerases can add 400 nucleotides per second. That's pretty damn fast. And they have an error rate of only 1 in 10 million base pairs. So that's pretty good. That's because they have what's called proofreading ability. They can remove the wrong nucleotide and replace it with the correct one if a wrong one's been put in. They have exonuclease activity, which means they can remove the nucleotides in the opposite direction from which they were laid down. They can only be laid down 5' to 3'. Exonuclease means they can remove them 3' to 5'. Also have endonuclease activity, which means they can come in and cut the sugar phosphate backbone to pluck out individual nucleotides that are not the right ones. So this shows you there can be proofreading at the level of nucleotide selection. So here the DNA polymerase molecule, the orange ball there is proofing which nucleotides are being brought in, making sure it's the right one. So here they're bringing in A, but an A doesn't pair with a G as C does, so it kicks out the A and has a C brought in instead, the correct one. They can also do proofreading by mismatch repair. Here the A has already been stuck in there. It doesn't pair with the G, so it's bulging out a little bit. So anyway, the DNA polymerase molecule, again shown by the orange ball, can come in, pluck out that A, and replace it with the correct nucleotide, a C in this case. So these are three ways that DNA polymerases can proofread, and this cuts down considerably on mutation rates. So this table discusses the enzymes involved and other molecules in DNA replication in prokaryotes. So if you can tell me what's on this table, you'll do fine on the exam. On to different structures that DNA molecules make while they're replicating. Most common one in prokaryotes is the theta structure. Sometimes there's a rolling circle where one strand is replicated linearly and one circularly. There's a displacement loop where you have the origin of replication in two different spots on the two DNA strands. And then for us eukaryotes, it's linear. So there are these four different DNA structures made during replication. So we've already discussed theta, and we'll discuss the other three now. This shows theta, rolling circle, and linear from your little table in your textbook. So here's the theta model or theta structure, like the Greek theta, kind of an oval with a line through the middle. The rolling circle is used by plasmids, some strains of E. coli and some phages. And basically what happens is the leading strand is synthesized circularly and the lagging strand is synthesized in a linear fashion. And here we see the leading strand in red there being synthesized from the origin. The lagging strand is now kind of unwinding and will be released kind of like a tail and be copied when it is linear and then made back into a circle again. So here we see that. Here's the rolling circle model here. Here are those two sides. Two slides start to finish. Mitochondrial DNA, that's what mtDNA stands for, the DNA molecule in the mitochondrion. Each strand has a separate origin of replication, and the origins are not in the same place, and replication starts at one origin and then proceeds until it hits the other origin, producing a sort of bubble or the D loop. So on the right, most diagrams are the easiest to understand here, and that red thing bubbling out, that's the D loop. Now, in eukaryotes, our DNA, of course, is synthesized linearly. We have at least 13 DNA polymerases. We have a slower rate, about 2,000 base pairs per minute, where in E. coli it's about 20,000 base pairs per minute. But ours is slowed by the histone proteins and the nucleosomes needing to be replicated, as well as the DNA that's wrapped around them. And of course, unlike prokaryotes, we have several, in fact, many thousands often, origins of replication. And of course, our replication stops at the telomeres, the ends of the chromosomes. So here's our origins in yellow there, showing leading and lagging strands, and how replication proceeds at equal rates in both directions away from the origins. You will see three origins 1 and 2, replication started at the same time, and 3 it started a little later. Then eventually those replication bubbles will all kind of meet up together, and our new strand is the red one, old strand is the black one. You see those slides all together in one. This shows you a photomicrograph of bidirectional DNA replication. So the black areas are the newly synthesized DNA there, probably labeled, it's a radiograph, so it's labeled with radioactivity. And then as time goes on, you can see the blackness spreading, and it's going to all coalesce together at some point. Here are 4 replication bubbles for replicating Drosophila DNA. A is the biggest one. B, C, and D are all smaller, indicating they started replicating later. The little black arrows point to the replication forks there, and eventually, of course, they will all sort of fuse together. This shows a eukaryotic replication. We have to copy the nucleosomes as well as the DNA, and that, of course, slows the process down. With regards to enzymes used in eukaryotic DNA replication, there's an alpha, a beta, gamma does the mitochondrial DNA, delta does most of the replication per se, leading and lagging strand synthesis. Epsilon, you don't really know, and there's 8 additional enzymes mostly involved in repairing damaged DNA. And this is a table listing what we think we know about them. And so replication proceeds until we hit the telomeres, which are at the ends of the chromosomes, and then things get a little bit different. So our telomeres contain the sequence TTA-GG repeated many, many times, and the DNA polymerases cannot synthesize a lagging strand of the telomere. So a specific enzyme called telomerase does that. It contains RNA, and it extends the 3' end of the by RNA templated DNA synthesis, it's called. So this thing that looks like a yellow hot dog is the telomerase, and the green thing in the middle of it is the RNA, showing you how it synthesizes the telomeres. And another step in that process, those last 2 slides combined as one, showing the whole process of telomere replication. A little close up on what's going on at the ends there. And that is the end of DNA replication.